Welcome to the Biology 182 Evolution series. This video will provide information on how populations evolve. At the end of this video, students should be able to identify if a population is in equilibrium or evolving. Populations are organisms of the same species living in the same habitat or ecosystem. The individuals within the population all have individual alleles that contribute to the population's gene pool. The gene pool is the sum of the total genes available to make up potential offspring for the next generation's gene pool. Population genetics is the study of the variability and changes within a gene pool. Because populations are dynamic, they can change in allele frequency, location, and size from one generation to the next. Allele frequencies are the prevalence of an allele in a population. This can change from one generation to the next. Remember that an allele is only one option for a trait that's being expressed. So an allele for freckles can be represented by the capital F for freckles or the lowercase f for no freckles. In order to determine an allele frequency, we combine the number of copies of one particular allele and then divide by the total number of alleles in the population. Remember that all the alleles in the population or gene pool total 100% of the alleles. Looking at an example, there are 8 individuals in each population. For population 1, the number of big A alleles is 4 out of 8, and the number of big B alleles is 4 out of 8. These alleles are in a 50 to 50% ratio, or 1 to 1. It also can be written as 0.5 to 0.5. In population 2, because there are 2 out of the 8 that are A and 6 out of the 8 that are B, we can see a ratio of 25% A to 75% B. We can easily discern population 3 at 100% A and population 4 at 100% B. Let's try diploid organisms. The total alleles in a diploid organism is 2 times the number of individuals, since they have 2 copies of each allele, 1 on each chromosome. To determine the number of freckle alleles, you would combine all the big F's, so that it would be 2 times the number of individuals that are homozygous for freckles, and 1 times the number of individuals that are heterozygote. Then you would divide by the total number of alleles in the population. In this example number 1, we see there are 7 warthogs. Of the 7 warthogs, 2 are homozygous dominant, 3 are heterozygous, and 2 are homozygous recessive. This means that there are 2 big B, so that is 4 big B alleles. There are 3 big B, little b, so there's another 3 big B alleles for a total of 7 alleles. The total alleles in the population is 14 because there are 7 organisms, and they are diploid. 7 out of 14 is 50%. To determine the small b allele frequency, we would combine the 3b from the heterozygotes and the 4 small b from the homozygote little b phenotype and genotype. This gives us 7 little b alleles. Again, we are at 50%. To check this, we add the 2 frequencies together. 0.5 and 0.5 equals 1. In example 2, there are 3 different genotypes: big G, big G, big G, little g, and little g. There are 10 rabbits. 8 are gray. 3 of the gray are homozygous dominant. Calculate the allele frequency for big G and little g. We know there are 10 rabbits in the population, and they are diploid, so that gives us 20 total alleles. We know that 3 of the rabbits are homozygous dominant, which gives us 6 total big G alleles. That means 5 of the gray rabbits must be heterozygote, which gives us another 5 alleles. This makes 11 out of the 20 alleles being big G. 11 out of 20 is 55%, or 0.55. For little g, we have 2 homozygous recessive alleles, so 4 little g alleles, and the 5 heterozygote little g alleles, totaling 9 small g. 9 out of 20 is 45%, or 0.45. To double check our work, we have 0.55 big G plus 0.45 little g equals 1. One way scientists represent allele frequencies is with the formula P plus Q equals 1, where P is the frequency of the dominant allele. Q is the frequency of the recessive allele. Hardy, an English mathematician, and Weinberg, an English physician, derived this formula to relate allelic and genotypic frequencies together. In this case, when looking at the rabbits, we could have just subtracted the 0.55 from the big G from 1, giving us 0.45 for the recessive small g allele. The genotypic frequency measures the occurrence of each genotype in a population. Each genotype in a diploid population will have 2 alleles. So if we look at flower color and say that the trait F for flower, capital F would be the dominant allele, and lowercase f would be the recessive allele. We know homozygous recessive will be little f, heterozygote will be big F, little f, and homozygous dominant will be big F, big F. When looking at the Q plus P equals 1 formula, P is represented by the big F, or the dominant trait. Q is represented by the small f, or recessive trait. Big F, frequency of homozygous dominant. Little f, frequency of homozygous recessive. So to find these amounts, we have to multiply big F times big F to get that frequency of homozygous dominant. We have to multiply little f times little f to get that frequency of homozygous recessive. And we must multiply big F times little f to get the frequency of the heterozygote. Because in a normal population following traditional Mendelian genetics, we would have 1 big F to 2 big F to 1 little f when we look at a homozygous dominant times homozygous recessive true breeding cross, we would expect the 1 to 2 to 1 ratio. Because of this, we need to multiply the heterozygote big F times little f times 2 in order to account for both the 2 intermediate genotypes. The Hardy-Weinberg equation reads P squared plus 2PQ plus Q squared equals 1. This will give us the genotypic frequencies for the F1 generation. If we look at the example of incomplete dominance in flower color here, we can see that the flowers are represented by big R for red, WW for white, and RW for pink. The parentals that were crossed were a WW white and an RR red. The Mendelian expected would give us the 1 to 2 to 1 ratio, 1 red to 2 pink to 1 white. If our parental allele frequency from the original gene pool is 0.7 for big R and 0.3 for W, how would we find the frequency of red? Well, we would use the 0.7 from the big R and multiply it times itself. 0.7 squared is 0.49. To find the PQ, we would multiply 0.7 times the 0.3. However, the chance of getting the heterozygote pink flower is 2, not 1, so we must multiply the 0.21 times 2, which yields 0.42. Finally, we have to multiply the 0.3 from the white, square it, so multiply it times itself to give us the frequency of the white, which is 0.09. When we sum 0.49, 0.42, and 0.09, we get a total of 1, which supports the Hardy-Weinberg equation. Hardy-Weinberg is a prediction of allelic and genotypic equilibrium within a population. This means the population cannot be evolving. The allele frequencies and genotypic frequencies must stay the same from one generation to the next. This means that random mating must be occurring. The population must be large enough to avoid random chance anomalies. There can be no migration from organisms into the population, and natural selection cannot be occurring. When populations are not in equilibrium, they are evolving. Microevolution is the change in allele frequency from one generation to the next. Students should be able to differentiate between populations that are evolving or in equilibrium. Students should be able to calculate allele frequencies. Students should be able to calculate genotypic frequencies. And students should be able to understand the rules required for Hardy-Weinberg equilibrium.